All right, folks. Well, this is a special episode of Healthcare Unfiltered because I get to talk. Well, I always talk to smarter people than me, but this is really an interesting episode because I have two scientists who are going to simplify a complex field that literally drove me crazy and made me um, very self-aware of how little I knew when I was a fellow at Northwestern, because I would get these reports, and at the time, long time ago, by the way, it wasn't probably as advanced as it is right now. We are going to delve into the world of cytogenetics, the world of cytogenetics and cancer care. And to do this, I am joined by two amazing guests that they will introduce themselves in a little bit and tell us a little bit what got them into this. And we're going to talk about what it is, what it means, and how it became even mainstream in cancer care. So I'll start with Linda, Dr. Baughn, who is joining me from Minnesota. We are taping this episode for context somewhere in the end of 2022 in December, although it is going to air in 2023. And the reason I say that is because whoever's going to watch this show, if they're watching when the sun is rising and it is no longer cold, they will understand why Linda have two sweaters on, because it is probably freezing in Minnesota. So Linda, a little bit introduce yourself, and then we will move on to Yasmine, Dr. Akkari.
Yes. So thank you, Shadi, for having me. I really appreciate it. It is about a week after ASH. It's really cold here in Minnesota. I think the high was -5 today. So I do have just one sweater, not two. I do have heat in my house. But yeah, so I am a cytogeneticist and molecular geneticist at the Mayo Clinic. I've been at the Mayo Clinic since 2015. I received, or I guess by way of introduction to my background, I received my PhD in 2004 from Tufts University, where I became really interested in cancer biology. So I was working in a laboratory where we were trying to understand how the B-ABL oncogene caused B-ALL in mice. Because I didn't want to only study cancer in mice, I went on to do a postdoc at Cornell in New York City. And there I became very interested in multiple myeloma research. I continued that postdoc for 3 years and then actually did another postdoc where I was looking at trying to understand how activation-induced cytidine deaminase, which is AID, is important for class switch recombination and somatic hypermutation. Now, I did a postdoc 6 years, but the reality is that my husband, who's also a scientist, got a job in Minnesota. So we moved as a family to Minnesota. And in Minnesota, I obtained a non-tenure track research assistant scientist position. And this is, I was grateful for this position, and I was in this position for 2 years, but I had some concerns about my viability of the fact that it was really not a tenure track position. So completely serendipitously, I attended a faculty meeting where the cytogeneticist at University of Minnesota mentioned that they had an opening for a fellowship opportunity. At this point, I'm in my mid-30s, and I had always imagined that I would be running a research lab. And the thought of going into a fellowship program was really scary for me, but I just took the plunge. I did cry a little bit because I was really nervous about, I knew nothing about this kind of career path because I wasn't told anything about this with a PhD. So it was a 3-year program, and I loved it. I loved it so much. I was so immensely grateful to have the opportunity to do the fellowship program. I completed that in 2015 and joined the Mayo Clinic, and I've been at Mayo Clinic since then.
Well, fascinating journey. And we're going to talk about that, what it means to be a cytogeneticist, because I think some of the listeners may not understand what it actually means. So we need to clarify that.
Yeah.
Yasmine, how about your journey? How did you get to where you are right now?
So Linda's very brave and talked a little bit about timeline. I'm not going to talk about timeline because I'm a lot older than Linda. So I actually, I came from Lebanon after having done a Bachelor of Science in Lebanon, and came to Ohio State for my PhD in molecular genetics. I did a postdoc on Fanconi anemia, and at that time, this was my first encounter with a human disease, as opposed to just the basic research on like mitosis, right? And then, I wanted to have a child and left work a little bit, and then came back to Fanconi anemia. And then also just by conversation with my mentor, he said, why don't you do a clinical genetics fellowship? And I'm like, what is that? And that's how that door was open. So very parallel to Linda, it was really a serendipitous find. And the reason why this is really important is because I think we are producing a lot of PhDs, much more than academic physicians may be able to accommodate. And there is a huge clinical path out there that needs this kind of expertise and need this kind of thinking and troubleshooting that we both would kind of benefit, right? And this is my kind of energy into talking, about this. So right now I am a clinical cytogeneticist and a clinical molecular geneticist. I've also done PhD medical genetics, which allows for genetic counseling. And I do work as a senior director at Nationwide Children's Hospital in Columbus, Ohio. So the journey, and I think the message here that we would like to relay is that there aren't enough of us. Shadi, I'm sure you are very aware that genetics or laboratory genetics now touches every aspect of medicine, whether it's cancer or pediatric or prenatal. And I think, the need to have experts in laboratory genetic testing has increased beyond our ability to recruit to the field. And that's why we're here.
That's interesting. So let's step back a little bit. Linda, when you say I'm a clinical cytogeneticist, that sounds like a foreign language to somebody who is listening, who is not, what does that actually mean? What do you do every day?
Yep. Yeah, that's a good question. So it was a foreign language to me too. In my PhD training, I didn't actually understand what this field means, but I can try to explain it to the listeners. So cytogenetics is the understanding of chromosomes. And the need to understand the chromosome structure really is important for many different areas of medicine. In my specific area, I am focusing on heme. I love heme very much so, and specifically B cells. But I can explain essentially what the day-to-day activity is. So when I'm on a clinical sign-out day, so for example, a lymphoid in our laboratory, we divide up the work in terms of content areas. So for lymphoid, we would be then receiving all of the specimens that have been sent for example, a lymphoid malignancy, a high-grade B-cell lymphoma, where we would need to look for a MYC rearrangement, BCL2 and BCL6. So we would be performing FISH testing, which is fluorescence in situ hybridization, where we have DNA probes that are complementary to specific regions of the chromosome, of the genome, that are fluorescently labeled. So we're specifically looking for example, a MYC rearrangement where we would have a split of a probe, or if we're looking for, let's say, a MYC IGH rearrangement, we're looking for fusion of MYC and IgH. In the lymphoid bin work, we also look at multiple myeloma. And in the context of multiple myeloma, we're looking for standard risk or high risk abnormalities. So we're able to identify whether a patient has an 1114 multiple myeloma that maybe has a TP53 deletion. And that kind of information will help inform the clinician the kinds of treatment, and at least the prognostication for that specific patient. While I do love heme, I do also occasionally perform the interpretation for a test called chromosomal microarray. We do this test also in the context of heme malignancies. But for example, for chromosomal microarray, this is the first tier test for an individual, often a child, that has an intellectual disability. So here we're looking genome-wide at copy number changes. And when we identify a significant copy number change, our job is to write an interpretive report that will then be given to the clinician and patient. So this is essentially the day-to-day in terms of the clinical interpretation, but there are other aspects of the job. For example, like many jobs, we also have meetings. We meet with our team, our clinical team. We also discuss aspects of assay validation and also just management of the laboratory. If there are any issues that come up, we need to deal with those and figure out how we're going to troubleshoot various clinical laboratory issues.
So Yasmine, is it fair to say then that it is just broad strokes? One is performing the test itself, and two is interpreting the test. You do both, performing and interpreting?
Yeah, so Shadi, if I take just a little bit of a step back, I mean, just for the listeners, as Linda mentioned, any alteration in those chromosomes, one copy from the chromosome comes from the mom, one copy from the dad, any alterations has been documented to cause disease, right? So whether on the prenatal side or the pediatric or the cancer, right? And the science of cytogenetics is really responsible for detecting those alterations. So to answer your question, I think you're right. Our profession has two sides. Well, actually, I want to say it has three sides. One is the actual technical aspect. So doing the test on the bench in the lab, right? And most of the cases, laboratory directors such as ourselves are the ones who are interpreting the results that come out of a team of genetic technologists that do the wet bench work. So the data comes to us, and then we put it together in an interpretive report, because the data comes as raw data, right? If you just put it immediately into the hands of the physician, it's very, very ununderstandable, right? There is a lot of nomenclature, there's a lot of technicalities. And so we take that data and we put it in an interpretive report, meaning, what does that mean? That alteration in the chromosome, what does it mean? Does it have diagnostic significance? Can we diagnose this child or this patient with a certain disease? Second, is there a prognostic factor to that test? And third, are there any therapies related to that particular chromosome alterations? So the role of the technologist is to actually do the test. Our role is to take that raw data and put it into interpretive report.
Makes sense. But let me just piggyback on this. These are, again, for listeners, this is a blood test, saliva test, bone marrow test. What is the actual specimen that you're doing the test on?
All of the above.
It really depends on the indication. So for a patient with multiple myeloma, a bone marrow biopsy is typically performed, and we receive the bone marrow. We also work very closely with heme path with their heme path colleagues that will do, for example, flow cytometry or other types of testing. And another aspect to the work is integrating the information from different areas. So for example, heme path, taking a look at the various results that they receive and how we can implement that into our interpretation to provide the most helpful information for the clinician and the patient.
So the other specimen is tumors, tumor biopsies, brain tumors, solid tumors, lung.
So basically, what you're doing is you're taking whatever is sent your way to the lab from the cancer center or from the pediatrics or from wherever it is, and your job is to identify if there's some chromosomal breakage in whatever you got sent. Okay. So what Linda told me earlier, Linda, you mentioned something called FISH, I believe. You're trying to allude to this, and I'm gonna assume that a lot of listeners don't know what you mean by FISH. So you're gonna simplify that to us because there's, you know, how do you find out about whether there's a chromosomal breakage or not? So, you know, maybe take us through this, and then Yasmine, after Linda finishes, maybe you can explain why this is even important. Like, who, you know, you mentioned a little bit about prognostic or therapeutic or other aspects, but illustrating this with examples, I think, will help listeners understand.
Yeah. So FISH is one of the most common assays that we in any cytogenetics laboratory is performing. So in a very simplistic way, FISH is, again, fluorescence in situ hybridization. So a specimen comes to our laboratory, and let's take bone marrow in this example. There are 3 main types of FISH tests, but they all require receiving the sample, fixing the cells, creating fixed cell pellets, dropping onto glass slides, denaturing, so the DNA, so we're looking at DNA alterations. The DNA, the double helix, has to be split so that the complementary FISH probe that is targeting a very specific region of the genome can bind. And those FISH probes have a fluorescent signal so that when you look under the microscope, you can actually look at individual cells and the kind of FISH abnormalities that you see. So let me explain the 3 different kinds of FISH probes.
But Linda, with FISH, you kind of know what you're quote unquote fishing for.
Yes,
right? Like
exactly.
You kinda, like, it's not really, I'm gonna see what happens. Like, you really have to say, I'm gonna look for this particular abnormality and I'm gonna see if it's there or not.
That is absolutely correct. And that is a bit of a limitation, because if you don't know what you're fishing for, you just are not going to be able to find it. So you have to have kind of an idea of what to look for. So again, going back to myeloma as the example, there are specific recurrent genetic abnormalities that are associated with a different disease outlook. And I mentioned 3 kind of main types of FISH probes. So in one simplistic way, you can have a FISH probe that targets the immunoglobulin heavy chain gene that is commonly associated with a translocation. And you can have 2 FISH probes that are labeled with a different color. And you're basically looking for a split of this FISH probe. And so that tells you, in a normal circumstance, you would have just 2 fusion signals. But if you have a split, you have a red and a green that are separated from one another. The other type is dual fusion. So in a normal situation where you would have 2 genes that are on different chromosomes far away from each other, but in this situation, you know that in cases where there is, for example, an 11-14 translocation, cyclin D1 that's on chromosome 11 happens to be right next to IgH, immunoglobulin, that's on chromosome 14. These fish probes have different colors. So when they are right next to each other, due to a chromosomal translocation, it shows up as a fusion. And then the last of the three types of fish probes is an enumeration. So we're essentially just counting dots, I guess, to be simplistic. And we often look for example, a TP53 deletion in a patient with myeloma. In a normal circumstance, you would have two normal copies of chromosome 17p, where the TP53 gene is located. But if you only have one copy, you know in that individual that there is a deletion of TP53 because the fish probe is sitting on TP53. So I hope that the listeners understand a little bit about what FISH is. Again, it's a very targeted type of assay where we use DNA probes that go directly to a specific region of the genome. And we're looking for copy differences in the amount of the FISH probes, or even a movement of the FISH probes from one location to another.
But Yasmine, don't they also sometimes do regular cytogenetics without fishing for anything? How is that different?
Yeah, so it's really interesting because cytogenetics, Shadi, is a science, right? It is the understanding of chromosomes. Within cytogenetics, there are several techniques. Just like under molecular genetics, there are PCR or sequencing or southern, blots. Under cytogenetics, there are many kinds of technique. FISH is one technique that we call, it's a little bit biased, right? Because like you said, you have to know what you're fishing for in order to fish. Whereas there are other like chromosome banding, just G-band analysis where you look at the karyotype, right? That's an unbiased view of the genome. You just look at the whole cell, the chromosome complement of a whole cell, and you see what is missing, what is translocated, what is deleted, what is duplicated. That is an unbiased look at the genome. And from that—
But how do you do that? Like with the fish, I think Linda explained you have these probes and all that stuff that they light up. When you want to look at the whole genome, how do you actually do it?
So you have to take the specimen that comes to the lab. You have to make it divide, meaning you have to have the cells enter mitosis in culture, in vitro, right? Like in vitro, that's right, in vitro. But out of the body, you put them in a beautiful kind of liquid that is very rich, a media, and you allow the cells to divide in culture. Then you take those cells, you drop them on glass slides, and you hope that you have arrested some of these cells in mitosis, right? The cell goes through a cell cycle. You want to arrest them in mitosis where the chromosomes are condensed. When the chromosomes are condensed, they're visually attainable, so we can see them, right? As opposed to a soup of DNA like spaghetti. When we put them on when you drop these mitotic cells on a slide, they all kind of stay in a way, and there are techniques to do that, which I'm not going to belabor, but they're basically a way to keep each chromosome component of a cell together. So you can count 46 chromosomes in a human cell, and then you align them homolog by homolog by staining them with a specific stain plus trypsin that allows for banding. What does that mean? What does banding mean? Banding is a pattern. Chromosome 1 has a banding pattern that is very different from chromosome 2 banding pattern. So you have chromosome 1 from mom and dad, you align them, and you see, okay, are they intact? Are they the same? Is there anything deleted, anything duplicated, anything translocated, anything inverted, anything inserted? All these chromosomal abnormalities can be seen by looking at that G-banded cell. So the trick of it all is that this is a technique that has been in use for more than 50 years. Is it the best resolution possible?
50? Did you say 50?
50. Yeah. Banding of chromosome, it's been since 1971 by Kasperson. Now when we look at it, and this is why we would love to address the issue that cytogenetics is dead. The science of cytogenetics cannot die. Techniques can
But
evolve.
who's saying the cytogenetic science is dead?
A lot of people are saying that.
Mm.
I think because there have been, I mean, since the sequencing of the human genome, right? When it was completed, I think a lot of people thought that we can really now, the tell all is going to be sequencing. So understanding the AGTC sequence of the genome, right? And I think in a lot of ways, we have come to an amazing progress in understanding the sequence of the human genome and how disease comes up. But the science of understanding chromosome structure, not the sequence, is really kind of taking a back burner.
Mm-hmm.
And it's hurting the field because young people hear cytogenetics is dead. We're never going to do cytogenetics anymore. We're just going to sequence. Is naive and really is kind of shortening the expertise in our field.
So let's step back a little bit because I want to get back to this. I think that's a very important point that you bring. There are obviously the descending view about cytogenetics. So I'd like to really delve into this just a little bit to better understand that view, although we don't have anybody on this show to represent that view. But how long does this process take? So if I send you a specimen for fish, Linda, and if I send you a specimen for regular banding, Yasmine, how long do you need to do? How long does it take you, Linda?
So for fish, actually, it's fairly quick. So, if we're comparing to something like next generation sequencing, we can be much quicker than that. So our quickest fish assay, which is involving PML RARA and also BCR-ABL, we can complete that work within 2 hours. So we can result out. If a specimen is coming into our lab in the morning, the clinician can get that result in the afternoon. So those are highly prioritized fish tests. An acute leukemia it has to be overnight. I mean, we need to have that result ASAP.
And then for regular banding, Yasmine?
I think for regular banding, depending on the specimen, so let's take a bone marrow. I think you have to culture it for 24 to 48 hours, depending on the disease you're looking for. I think within 4 days or 5 days, because we also have to allow for analysis, which is kind of an involved process and takes a lot of expertise. But I think within 4 or 5 days, we would issue a report.
So why do I need regular banding though? I mean, it seems like fish is going to tell me exactly the chromosomes I'm looking at. So does this nullify the need for regular karyotyping?
Well, no, because fish again is targeted. What if you don't know what you're looking for?
Yeah, but my question is, are there situations like, I presume in leukemia, lymphoma, myeloma, you already know what you're looking for. Yeah. So what you're saying, Yasmine, is there are certain diseases where you don't know and you're just kind of looking at.
Right.
Okay. So Linda, take me through that descending view. I'm very intrigued by this. I honestly did not know that there is a descending view to the world of cytogenetics, but it's really important to understand that. So I'm sure you know people who have these views. What do they say?
Well, sometimes I also have those views too. So again, going back to myeloma, what we have learned, and not just myeloma, but other heme malignancies, we, as a cytogenetics laboratory, had started a NGS type test called mate pair sequencing, which we're not doing currently. But this was established within our laboratory probably around 2016. And it was essentially a less expensive version of whole genome sequencing. We used this technology and we realized that, wow, our gold standard fish test is sometimes wrong. So as an example,
is it wrong because technical difficulty, or the probe is wrong? Like, why would it be? It seems like it's too specific to be wrong.
It's wrong in some ways because of the biology. So let's go back to, let's say mantle cell lymphoma with an 1114 rearrangement. Often you would have a direct 1114, a balanced rearrangement, but in some instances, you may have a small portion of the immunoglobulin promoter enhancer inserted upstream of cyclin D1. Or, for example, we learned that MYC rearrangements are not always represented by an balanced an 814 rearrangement. You may have a small enhancer that is very close to MYC, where your MYC probe will be completely normal. It's not that there's a technical issue, it's that the biology is telling us that sometimes you can have a rearrangement, but not have a disruption of the FISH probe. So for a dissenting view, I do think that in the future, we will most likely be doing whole genome sequencing that will allow us the opportunity to see within the genome all of the structural rearrangements, copy number, and single nucleotide variants that will allow us a much more precise way to look at the genome, that the FISH technology, that will be better than the FISH technology. But one point that I do want to make is that if you look at a chromosome study or FISH, that's actually on a single cell level. But if you're talking about something like whole genome sequencing, typically it's bulk. And so the sensitivity is not going to be as great as if you were to look at a FISH test where we can identify 3 cells that have a BCR-ABL translocation out of, for example, 200 cells.
But Yasmine, I mean, it seems like the dissenting view is proposing that if I'm getting my information through NGS or through whatever other technology, and I'm not missing anything through that technology, then I'm just consolidating. I'm just doing one test versus doing this and that. I mean, I can understand that. I can understand that they're not saying it's not a good test or a bad field. They're just saying, we've evolved, we've progressed. Science has moved on from 1971. What do you say to that?
Yeah, no, I think that's exactly true, Shadi. I wish, so I'm going to elaborate a little bit on the concern. The concern is not that newer technologies are not better. They are. Whole genome sequencing right now can detect everything that a classical G-banding, classical karyotyping can, and perhaps more, because it allows us to really do high resolution. It also allows us to get sequence information, not just the big structural, abnormalities of the chromosome. The concern is cytogenetics is not karyotyping. Cytogenetics is the science of understanding chromosomes. And if we don't have that fundamental knowledge of understanding chromosome, interpreting whole genome sequencing data is going to be very challenging. And our concern, both Linda, myself, and a lot of cytogeneticists out there, is that we are diluting that knowledge, that interest in looking at the whole genome view of a single karyotype and visually understand the chromosome interactions, and then applying that knowledge to newer technologies. I totally agree with the fact that all technologies evolve, all sciences evolve. I think karyotype is also going to evolve, and it has. I mean, whole genome sequencing is extremely powerful. But from an educational perspective, it's still very important to understand the fundamentals of the science, which is how chromosomes move.
Yeah. So, Linda, the cytogenetic test, whether it is FISH or banding, or is this something available anywhere? Like, any hospital practice? I mean, how available is this test in small community hospital somewhere?
Mm-hmm. Mm-hmm. Yeah, I would say, I mean, I work in fairly mid-sized to large reference laboratory. I think it's fair to say that most large university hospitals do have a cytogenetics laboratory or a molecular genetics laboratory. Although I don't have data to support this, I have observed that there may be some smaller laboratories that are struggling a little bit more. I think that I am a little bit concerned that over time, this kind of testing will be focused more on the larger laboratories, especially when we do move to more expensive NGS type tests. It may be difficult for smaller laboratories to keep up with that type of, purchasing of capital equipment, assay validation, which is very expensive. But for the most part, at any given, even community hospital or community setting, a clinician should be aware of where to send the specimen for cytogenetic testing or have it performed in-house.
Got it. Yasmine, take me through the relationship between you, the cytogeneticist, this is the correct description, and the pathologist, right? Not the clinician, because as a clinician, I need you and I need your help and interpretation. I'll be the first to say that. But where does the pathologist fits in the equation, and specifically the molecular pathologist? We're seeing in pathology, there are some folks who are called molecular pathologists.
Yeah.
So do you go and meet with the pathologist and you go over the report? Do you just issue that? Does the pathologist have an input? Like what is the intersection between the cytogenetic lab and the pathology lab or the molecular pathology lab?
So I think I'm going to step back just for a second and say that in our field, in our kind of profession, there are two big buckets of work we do. One are the diseases that you are born with, and those are constitutionals. And this is where we do the pediatric and the prenatal. Those really we have no interactions with the pathologist, right? Because the specimens come to us, we test and we say, yes, this child has trisomy 21, right? Or Down syndrome, or this child has a deletion of chromosome 22 and therefore has a heart defect. Those are diseases that people are born with. And pathologists, really, there's very minimal interactions. When there's really, really valuable interaction is in the cancer field, right? Because this is where tissues come through pathology. We interact very, very closely with hematopathologists when we have a hematologic malignancy. We interact very much with the surgical pathologists when there are solid tumors that we have to look into to test. So, I have to say the pathologists do rely on cytogeneticists to see if there are any chromosomal abnormalities. Molecular pathologists and molecular geneticists, such as Linda and myself, we kind of work very closely in cancer. But one thing I do want to mention, because you asked a very, very important question, Shadi, about is cytogenetics available to everybody, right? And is whole genome sequencing available to everybody? And I do want to pause on this because this is really, in my mind, and perhaps I'm biased, this is about global equity, right? So I have the privilege to sit as the genetics expert on the WHO classification of tumors. And I know that you have many podcasts about this particular topic. When we sit together as a editorial board and say, okay, we're going to classify these tumors, and we're going to say, what are the essential molecular pathology tests that we need to do? We very much keep in mind that there are some countries who cannot afford to do whole genome sequencing. So we have to also say, you can do cytogenetics. It is a well-established technology. I mean, not technology, it's a well-established science. And karyotyping is a well-established, available technology within cytogenetics. So,
yeah. So I want to try to understand the how, Linda, or mean, how many of you are there? Like, where we talk about, like, is there a society of cytogeneticists? Like, I know there's maybe 10, 12,000 oncologists out there. I mean, do you have, is there 1,000? Is there 5,000? Are there 20,000? Like, how many of you are there? I'm trying to match the supply and demand a little bit. I'm trying to understand, is there a real shortage, or is there really a perception of a shortage?
Yeah. So, I can't tell you, Yasmine, do you know how many total cytogeneticists are there? But I can tell you that probably every year about 50 or so do get boarded in the American.
That's nothing. 50? 50?
Yeah.
That's nothing.
I think in the US, the latest estimate is less than 300 cytogeneticists.
So the entire country, the entire United States of America has 300 cytogeneticists.
Yes.
So, wow, I'm a little bit, I'm very taken aback by the number, because I'm presuming this is why there's a lot of send out tests, because most hospitals then won't have local expertise, no?
That's correct.
I mean, there's no way. I mean, if you have one per hospital, that only covers 300 hospitals.
Yes, that's true.
Yeah.
So how, I mean, something is missing here. It's either there's a missing link. Help me understand, how can we, if there's so much need for this, how can we only have 300?
Because it's centralized.
It's centralized?
It's in a way centralized. Yeah, I don't think every community hospital can really afford.
So they can send to Mayo, or they send to elsewhere.
Correct. I mean, Linda, am I correct in that? I think there's a lot of centralization of cytogenetics, and it's also, from a CLIA perspective, it is considered high complexity, right? Just like molecular genetics, it's high complexity. So they need to be expertise.
I think a bottleneck could also be, and this is not, a major area of expertise regarding the education, but there are a certain number of training programs, ABMGG, American Board of Medical Genetics and Genomics training programs, and the training programs may only have 2-3 slots per given year. And so that, does require resources. So some training programs may not have abundant resources to train 10 people, for example. So I think that could be a potential bottleneck as well. But there is centralization. There are very large laboratories, LabCorp, for example, that are receiving numerous, probably thousands of specimens per day to do this work.
But I mean, how many cytogeneticists does LabCorp employ? I mean, I'm trying to think, if the entire country has only 300, you could literally demand doubling your salary, and they're going to have to pay you.
Right, but I want to also clarify that less than 300 are actually active clinical cytogeneticists. It is possible that some cytogeneticists have gone into the industry sector, and they're not actively signing out cytogenetics reporting. Right. So I think that big laboratories do employ a lot, but there's also distribution of demand, right? Some hospitals specialize in prenatal and pediatric. Other hospitals specialize in oncology, and it's kind of divided between us.
Sure. So in the world of cytogenetics, in the last 10 minutes or so, in the world of cytogenetics, what do cytogeneticists disagree on? Or what are the points of disagreement or debates? Or, I presume that we all agree that there is value in knowing the chromosomal abnormalities. Some of them may not affect treatment, but they can tell you the prognosis. Some of them might affect treatment now that you have a little bit more targeted therapies that target a particular like the BCR-ABL, for example, when you see that it affects that. So I presume there's no disagreement there. What do, if I put two cytogeneticists on the podium for a pro and a point and a counterpoint, what would be top 2 or 3 things that there are arguments about, or are you all agreeing on everything?
I think that, and just speaking from experience,
Trying to put Linda
Yasmine
against Yasmine and Yasmine against Linda. That's my way.
So I think, like, for example, the size of the fish panels, we will probably disagree on that. We tend to have fairly large fish panels. I think that there are some that are much more conservative and will do only a limited number of fish tests. Other ones may not actually impact patient care directly, but may be informative for the clinician and family. I tend to view that more information may be better, but at the counter argument would be, for example, like if maybe it's not totally necessary to move to a whole genome test for certain malignancies when you get all of the information from a really fairly robust fish panel. So why spend, thousands of dollars getting additional information plus the risk of an incidental finding that the patient may not really want to know when you can get all of the information from a good fish panel? So I think that there is that disagreement. And also, we hear this often that cytogenetics is dead. And this is something that Yasmine had brought up before. I think that there is this perception that it's kind of an archaic kind of methodology. But as I mentioned, the chromosome study, the karyotyping, is the first whole genome analysis of a cell on a single cell. And it's really exciting. And a lot of information can be captured. You don't need, a fancy whole genome sequencing test every single time. It's like, if you need to go to the grocery store, you don't need to drive a Lamborghini, right? You can take a bike and it could be fine. It could be good enough. So I think that it's a lot of kind of economics and the value, because ultimately somebody has to pay for an expensive test. And I think we have to be very careful with the resources that we have. So I think those are the things that I think about. I don't know, Yasmine, if you have other arguments.
I agree. Shadi, to your point, I think there isn't a huge amount of disagreement amongst cytogeneticists. The thing that I would the things that I struggle with, I think it's probably my favorite question, right? I struggle with making sure that we are offering the right test to the right patient for the right disease at the right time. That is kind of my slogan, right? And that has a lot of nuances. I struggle in the status of a PhD clinical provider not being able to bill as much as my MD colleagues can, just because I have a PhD and they have an MD, although we both do the same interpretive component of a reporting, right? Those are the challenges that laboratory geneticists, PhDs, whether cytogenetics or molecular genetics, really struggle with. What test to use for the right clinical, situation? Why can't we have the same billing rights as an MD?
So my last question, one for each one of you, is, Linda, where do you want cytogenetics as a field be in 5 years from now?
Well, I would like to know what I hope in 5 years we will know what is the right test to use for various malignancies. So in the context of AML, I think our FISH panels are robust and we don't need whole genome sequencing. But there are other malignancies where we do know that we're missing certain lesions using FISH testing. I hope that in 5 years, we can implement a a good whole genome sequencing test that can detect all of the different kinds of chromosomal abnormalities that we're interested in, and that it could actually improve patient care. This is what my hope in 5 years can be for cytogenetics. I think ultimately, you know, we probably, while, as Yasmine mentioned, cytogenetics is a science, I think that there is some confusion about cytogenetics versus molecular genetics. Within our laboratory, we're essentially just the genomics laboratory, so we're kind of combined. I hope in 5 years that those silos are kind of taken down and we are functioning as a genomics laboratory in a unified manner.
Yasmine, what questions I should have asked that I did not ask?
No, I think this was an amazing discussion, and we really do appreciate the opportunity to talk to you about things that we're certainly passionate about. I think if you were asked to ask me what would I love to see in terms of cytogenetics in 5 years, I think I would like to make sure that we recruit more young minds to this field. Make sure that they understand that science is still valuable for patient care, and also, ask them to join in this profession. We have the privilege of doing patient care on a regular basis as PhDs. So we use our expertise in troubleshooting, in the hypothesis-driven testing, right? To do very direct patient care. And I really hope that through your podcast and other venues, to let them know that there is a huge profession out there that could be very fulfilling, and to keep trying to understand all aspects of genomics, cytogenetics and molecular genetics, so that you can arrive to the right test for the right patient. So that's my hope, is to recruit more people.
I'm really fascinated by this. I think for listeners, hopefully they understand the importance of this, because there are so many diseases that we need to understand the cytogenetic profile. But I hope they also understood the level of training that you go through to get where you are. Linda did 2 postdocs, not only 1. And I also hope they realize that you're obviously a very important integral member of the team. I remain flabbergasted by the number of few hundred cytogenetics, given how big the country is and the number of hospitals and how important the test is to make these decisions. I can understand that, I mean, folks who might say we do the whole genome if I get all the information, because at the end of the day, I think doing the whole genome has become cheaper every year. I totally understand. It was very expensive at some point, but you could do it significantly cheaper. And who knows? I mean, I don't know how much the cost of cytogenetics, but there are insurance. It's a whole new can of worms. We can't really open it. But I've been really, thank you so much for coming on the show. I really appreciate it, and I hope that we can share this broadly with everybody who listens and beyond.
Yeah, thank you so much for having us. I guess if I could have one last message for the listeners would be that, if you happen to be a PhD student or you're in a postdoc, to really consider this as a really exciting and wonderful career. It provides a lot of job stability. You interact with amazing colleagues. I truly feel so grateful that I serendipitously fell into it. It's one of the probably luckiest experiences in my life. And it's just great. So I think that the listeners could also, don't be shy to reach out to us too directly. And we would be happy to discuss the career further with them.
Well, they can connect with you through the website, through the podcast, through our YouTube. Yeah. But Drs. Baughn and Akkari, thank you so much for coming on Healthcare Unfiltered. And cytogenetics field is not dead and will not die, and it's going to stay alive as long as we are alive. That's how it's going to go.
Exactly.
Thank you so much for having us. This was so fun. Really appreciate it.